Good morning to all of you. Let us continue with the chapter molecular basis of inheritance. In my previous videos, I have started molecular basis of inheritance. Today, let me explain the topic called packaging of DNA, which is one of the most important topic. Here, you can expect the 1 mark, 2 mark, 3 mark, rarely 5 mark question. So before I begin with this topic, let me tell you, my dear students, how the questions will appear. Okay, the definitions will come. What are these terms? What is nucleosome? 1 mark, all the definitions. Then 2 mark, they will ask you to draw this nucleosome structure. Okay, or else in sometimes 3 marks they will be asking. 2 or 3 marks diagram. A rare question of 5 marks, in that they will ask you to very rare explain the packaging of DNA. So at that time, with the diagram, with all the definitions and the process of packaging, you have to explain it. Okay, my dear students. So now, let me tell you, before I start the packaging of DNA, let me tell you some of the things which are essential to know to study this text. Basically, if we talk about the DNA, we are familiar with the DNA now, isn't it? That is deoxyribonucleic acid. Okay, where do you find the DNA beta? The DNA is found in the nucleus of the cell. In the eukaryotes or prokaryotes? In prokaryotes and also in the eukaryotes. But there is a difference in the eukaryotes and in the prokaryotes. You can see what I am drawing here is a circular DNA, double circular DNA. And if I say to people, this is double helical. Both are double stranded. But I can tell it as circular DNA. Okay, beta? What I can call it as beta? Linear DNA, which is also I can say both are double stranded DNA. Both double stranded. Why I am telling circular? Here there is no free ends. Are you able to see better? Free ends are there? No. Here free ends are present. That is the reason we are saying it as a linear DNA, double helical structure. Where do you find this type? You find in the bacteria. Okay? That is prokaryotic cells, shall I say? Yes. E. coli bacteria, do you know better? E. coli bacteria. This is a E. coli bacteria. In the nucleus, prokaryotes, they don't have the nuclear membrane. Eukaryotes, they have the nuclear membrane surrounding the cell. Surrounding the genetic material, not cell. Okay. So here, we are able to see it is a prokaryotic cell. For example, in the bacteria. Here it is a eukaryotic cell. It may be human cell. Okay. What is the difference you are feeling better here? Here in the prokaryotic cells, the DNA is double helical but circular. Double stranded but circular. Why? There is no free sticky ends are there. There it is a linear. There is a free ends are there. Okay, dear. So now here the nuclear membrane is not present, so it is scattered in the cytoplasm. Where? In the prokaryotes. That is example I have given bacteria. E. coli bacteria, we can take the example, isn't it? So here in the eukaryotic cell, the nucleus is covered with the nuclear membrane. Nucleus is covered with the nuclear membrane. That is what we are saying that eukaryotic cell. Modified than that of the prokaryotic cell. So here example we can say human cell. Human cell also I can say eukaryotic cells. So now where exactly the cells this DNA is located? If you see my dear students, this is the cell, eukaryotic cell I am telling about. and here it is a nuclear membrane. In that membrane, you find the chromosomes. Inside the chromosome, a chromatin thread-like structure that is nothing but DNA is packaged. This process in detail we are going to study here. Understood? What is the concept we are going to study?
Yes.
So you can find here different types of DNAs will be there. Yesterday in the last class I have said it. B DNA, B form DNA, Z form DNA, so many are there. A form DNA. So what Watson and Crick discovered, that is the B DNA, right-handedly coiled. Depending upon the type of coiling, double helical structure. Okay, ma'am? So now let us talk about the packaging of DNA. Do you know, I already repeated many times, the length of the DNA depends upon the number of nucleotides present in the polynucleotide chain. Isn't it? So, if you talk about the length of the DNA, a human DNA, a eukaryotic cell, the length of the DNA is 2.2 meter. 2.2 meter, this is more than this much. The way the girls are using, it is exactly near to that one. Isn't it? Is it so short? It is very lengthy. Isn't it? But how come it is possible to wrap that much of DNA inside a very minute cell? Even the cell is only minute, the nucleus is very smaller than that of that minute cell. Isn't it? So within that, inside the chromosome, it is packaged. How it is packaged? That we are going to study here in this chapter very clearly. Isn't it? So if you talk about this further, you can see that the DNA, when we are talking about the DNA, we have seen that it has a 34 Armstrong
length.
The length of each group. So what will be the difference between the two base pairs? Yesterday I had 3.4 4 Armstrong or nanometer also we can take. Okay, 3.4 nanometer. That is, we are taking 3.4 into 10 to the power of 9. Isn't it?
Minus 9.
Yes, minus 9 we are taking. Isn't it? So now, if you want to know whatever the DNA content, how many base pairs, and the number of the thing present in the what we call it as eukaryotic cell, what we have to do is we have to calculate, that is the length of the double helical DNA, isn't it? In a typical mammalian cell is calculated by multiplying the total number of base pairs into distance. Total number of base pairs into distance. So the total number of base pairs are 6.6 into 10 to the power of 9 base pairs. Okay, 6.6 into 10 to the power of 9 base pairs into this distance, that is 3.4 into 10 to the power of minus 9. So this is a typical mammalian cell. We can identify, that is how many base pairs at what distance. Okay, ma'am? So you can also calculate, that is even the E. coli, that is 1.36 millimeter, it is a distance. You can take the number of base pairs present and you can multiply and you can calculate, isn't it? So what it will be? We are just base pairs into, that is meter per base pairs, that is we are going to write like this. Okay, got my point, Peter? So now it has to be, that is the total length of the DNA is 2.2 meter. 2.2 meter DNA it is going to be, that is in prokaryotes, E. coli, the nucleus is not definitely present there. The DNA is not getting what we call it as completely present at one side, as DNA is negatively charged. You know that negative and positivity always attract with each other. Opposite charges attract with each other. Same charges repulse. Isn't it? DNA is negatively charged. Okay, DNA is negatively charged and held with some of the positively charged proteins. Prokaryotic cells, in the prokaryotic cells, what happens, you know? As a DNA is negatively charged, okay, so the DNA has to be held at one position because in the prokaryotic cells, the nuclear membrane is not present. So it is held with some of the proteins which are positively charged. Got my point? So here beta, the DNA in nucleoid is organized here, held by some of the positively charged proteins, forming a nucleoid. Did you remember in my previous videos I have said the location of DNA in the prokaryotic cell? Where do you find the DNA in the prokaryotic cells? Nucleoid. The negatively charged DNA combining with the positively charged proteins, and it does not allow the DNA to scatter whole in the cytoplasm as these positively charged proteins are held together. Okay? So now, what about the eukaryotic cells? If you talk about the eukaryotic cells, what is the thing happening here? Isn't it? In the eukaryotic cells, the DNA helix is going to be wrapped. DNA is a positively charged, and the protein DNA is getting wrapped over the protein, that is positively charged protein. We are able to see that. So it is going to be positively charged, wrapped around the positively charged proteins called histones. What are histones?
Positively charged.
Positively charged proteins. Why it is called as positively charged protein?
Because they hold it DNA.
They hold DNA, okay. Why it is said to be positively charged?
The DNA is negative, so negative is held by positive.
Okay. Protein is made up of amino acids.
Yes.
Made up of amino acid. This is one amino acid. This is one type of amino acid. There are totally 20 different types of amino acids are there. Among that, this histone protein is made up of these two types of amino acids, that is lysine and arginine. And these two proteins, the amino acids, are positively charged. Hence, this protein is positively charged protein. The histone is what is histone? Histone is positively charged protein. How you can ask me why it is said to be positively charged protein? Because it is made up of basic positively charged amino acids, that is lysine and arginine. So here DNA is wrapping surround the positively charged protein, that is histones, made up of lysine and arginine. Why it is wrapping? Because the DNA is negatively charged and the histones are positively charged. So always the opposite charges will be attracting to each other, right? So how the process will be taking place, we'll be seeing now. So as you know that, so here once again I can repeat here, DNA helix wrap around the positively charged proteins called histones to form a structural unit called as nucleosome. What is nucleosome? Double helical negatively charged DNA, or DNA, or you can write like this also. DNA is wrapped around the positively charged proteins around the positively charged proteins called histones to form a structural unit, many structural units, to form many structural units called nucleosomes. And see, one more time, one thing I wanted to make you clear is that here, see what DNA is doing, how the packaging is taking place in the eukaryotic cells, in the prokaryotic I said you. What about the eukaryotic cells? In the eukaryotic cell, the double helical structure of the DNA, which is negatively charged, is wrapping over the positively charged proteins. Isn't it? So when the negatively charged DNA is wrapping around the positively charged protein histone, it is forming many structural units. Many structural units, that is nucleosomes. Each we are calling it as nucleosome. So these nucleosomes combine to form a chromatin fiber. Chromatin fiber. Nucleosome से क्या बन रहा है? It is forming the chromatin fibers that is going to form, which appears like bead on a string. Beads are there. The beads will be there. The beads are how they are arranged. They are arranged in a thread or on a string. So how they are the nucleosomes when they are forming the chromatin? They are packaging the DNA and they are forming the chromatin fibers, isn't it? So how they are looking like? They are looking like the beads which are arranged on a string. Understanding? Getting what I am saying? Okay. So here, if you see the beads on a string appearance, then you can't see through your naked eyes. When it is present on the, when you observe under the electron microscope, you can find the structure appears like beads on a string. Okay, so actually if you see the structure, how it will be, the condensed chromatin fibers are further coiled and condensed at metaphase stage. Condensation, it is getting condensed, more condensed and condensed. During the mitotic cell division it is taking place, right? So the, what we call it as during the cell division, the copy, true copy has to be taken place now. In one of the class I have explained to people regarding that each of the cell is having the same DNA in our body. Isn't it? How it is possible? It is possible because of the cell division taking place, during which the genetic material, that is the DNA, is also copied. First class I have said this. Remember?
Yes.
So that cell division consisting of so many phases: prophase, metaphase, anaphase, telophase. Phases into there. During the metaphase stage, these chromatin fibers are coiled further and further. Continuously they are coiling and they are becoming condensed to form the chromosomes, isn't it? Packing of this chromatin to form chromosome requires some of the additional proteins also. Not only this, it also requires some additional proteins also. So such proteins are called non-histone chromosomal protein. In short, we call it as NHC proteins. Non-histone chromosomal proteins. Why we call it as non-histone chromosomal proteins? Because if you see, if you talk about is forming, right? Isn't it? First it will appear like bead on the stem, then later on during the metaphase again it is going to coil to form the resemblance, that is a structure so called as chromosomes. Understanding how the packaging is taking place? And this packaging along with the histone proteins also requires some of the other proteins which are also involved or are required for packaging. Okay, required for packaging, and that proteins are called as NHC proteins. What do you mean by NHC proteins?
Non-histonic.
Non-histone chromosomal proteins. Right, Beta? Histone proteins are involved in packaging totally because it is wrapped surrounding that protein only. DNA is getting wrapped surrounding that protein only. But here there are other proteins also are required. For example, you are able to see here. Here also one of the protein is there, which is not totally involved, but it is a part in packaging. Non-histone proteins, they are called as, it is non-histone chromosomal protein. In a typical nucleus, if you see, some of the chromatin are loosely packed. Okay, in the nucleus, if you see, you can find some chromatins are loosely packed. So such will be appearing light when we stain them, that will appear light stain, light color when we are applying stain. So they are lightly stained. Okay, lightly stained. Whereas the others are packed full, that is thickly it is going to be packed. So loosely packed will get lightly stained and they are transcriptionally active. ये क्या रहता है? Transcriptionally active रहते हैं. They are called as euchromatin. What they are called as? Euchromatin. And this region replicates early during the S phase. So this region, these are active in transcription process and are active during the transcription. It is very active, and it is, you know that during the S phase it will be active. There are some region of chromatin which are densely packed, which are packed very densely. Okay, and they get darkly stained. Dark stain when we apply the stain, and these are transcriptionally inactive. Transcriptionally inactive. Okay, and here they are active during the S phase of cell division, and these are called as euchromatin, and these are called as heterochromatin. Okay, heterochromatin. Two more questions most probably will be asked. Write the difference between euchromatin and heterochromatin. Okay, write the difference between euchromatin and heterochromatin. You have to write. Euchromatins are nothing but these are during the packaging, some chromatins are loosely packed and they get lightly stained, and these are active in transcriptional and are active during S phase of cell division. These are called as euchromatin. What about the heterochromatin beta? These are densely packed, tightly packed. Densely means tightly packed, and they Directly 2 mark question, write the difference between mostly asked question, or else what is euchromatin? What is heterochromatin? Okay, understood? So here late replicating replicates in the, it is late, it is fast replicating, it is late at the end of S phase. Okay, understood beta? Beginning of the S phase, it will be active. Start replicating at the beginning of the S phase. It is less active. Starts at the end of the S phase. Replicating starts at the end of the S phase. Understood? When you are supposed to ask, explain about the new packaging of DNA, you have to draw this diagram and you have to mention all those things, and it should also include the euchromatin and heterochromatin. Okay? Understood?
Yes.
Now, let us talk about. Now, there is a search for the genetic material. This is about the DNA. Structure of DNA we have studied, and we have also studied the packaging of the DNA. Now, let me explain you people the search for the DNA. Isn't it? Search for the DNA. Any doubt regarding it? Any doubt regarding?
Nucleosome it coils continuously.
Okay, search for the nuclear genetic material. Okay, now let me explain you people transforming principle. Search for the DNA genetic material. See, now I can say, if I ask you people what is the genetic material, can you tell me which is the genetic material? You can easily say. Now there are 2 genetic material, but among that 2, DNA is a genetic material in all prokaryotic cells, eukaryotic cells, bacteria, and also in some of the viruses. Whereas RNA is a genetic material in some of the viruses. Where is the exceptional case where DNA is not a genetic material? That RNA is a genetic material. Example HIV virus. Okay, but we know now DNA is a genetic material. We are safe. But earlier, before the discovery, after the discovery also, like it was like a question mark. What is the genetic material? Either protein, RNA, DNA. What exactly among these is the genetic material? Which is, now we can say. But earlier, if you don't know anything about the DNA, now we know genetic material is DNA only. You just don't think anything about, you don't know anything about DNA. If I ask you, can you tell me which is the genetic material? We have to search it. We have to do an experiment. We have to confirm it. Then only we have to say, then only we are able to see that confirmly either protein is a genetic material, or RNA is a genetic material, or DNA is a genetic material. So let us confirm with that, because we are assuming that we don't know what is the genetic material. So search for the genetic material. In that first experiment, that is transforming principle we will do. Okay, transforming principle. This experiment will come for 5-mark question. Okay, where you have to explain all the things what I am going to explain to people.